Hi, T. We'll start in a few minutes. Hey, Gwen. We'll start in a few minutes. This room is about using AI, machine learning, to design synthetic human gut microbiome. Our guest speaker, I just had the introduction room with him, and he'll be here in a minute. Hi, Gilbert. Oh, hi, Richard. How are you? Thanks for coming. Hi, Ethan. Hi, Birgel. Albert Hero, he will come in a few minutes. We just had the introduction meeting, and he will talk about his part of this study using machine learning to design synthetic human gut microbiome. And on the top, you see the link to the presentation. If you want to click on it and start checking it out. And in the chat, I added the paper link. It should be open source, so you should be able to access it. So yeah, we'll start in a few minutes. Hi, Denise. Hi, Mayor. How are you today?
Hi, Katerina. Hi, Gilbert. Hi, everyone. This should be an interesting talk. I definitely want to hear about what's going on in the synthetic gut microbiome AI realm.
Yeah, so our guest speaker today is the expert of the machine learning designing. Hi, Alfred is here. Hi, Al, how are you? We just had a meeting, so we were just talking 2 minutes ago. Um, Welcome, Al. Meet Gilbert, Dennis, and Mayor. Um, and yeah, thanks for coming.
Can you hear me?
Yes.
We sure
We can.
can.
Great,
Pleasure to
great.
meet you.
Good to meet
And
you
that,
as well.
thanks for coming here. I think this room is probably going to grow. I mean, this is an awesome title: Synthetic Human Gut Microbiome with AI. I am very excited to hear um, this topic for sure.
Great. Yeah. Well, there's lots of interesting things that revolve around the gut microbiome and around AI too. And this is a meeting of the minds. So you'll hear more about how we are making progress in understanding how the gut microbiome is effectively designable and Uh, using AI can be used to identify different types of species, bacteria in the microbiome that can accomplish the function that's desired.
Yeah, and are they like? I remember watching shows about this a while back, and it was fascinating how you can take some of the material from one uh, microbiome in the gut, and then put it into another individual, which provides them. It's almost like a medicine, right? Where you're
Yeah,
transferring from one person to another to heal issues. So that's fascinating. And then the whole, I guess it's a big problem with the antibiotics, where the antibiotics can go in there and kind of mess up the gut biome, right? And
exactly. Yeah,
yeah, so it'd be nice to hear about that, especially like what things to avoid, so we can keep our microbiome healthy.
yeah,
Which antibiotics are destroying the microbiome.
yeah. And it's not just keeping it healthy, but also for enhancing performance. You can add certain nutrients certainly to your body to give you enhanced strength or stamina or what have you. But the microbiome plays an equally important role, and so yeah, it's a very important component, one of the least understood of the sort of human host um, that maintains health and fights off disease and enhances our livelihood.
Awesome, awesome. Well, so is this your first day on Clubhouse?
It is, yeah, yeah. I'm not familiar with this venue. It's kind of nice being not having to share screen. As always, you'll worry that something's going to go wrong, technical difficulties, and so forth. The phone is just so much more reliable, um, and I think it leads to a somewhat less stressful experience. We'll see, of course.
Well, this is a great club. Like coming in, I mean, Katerina's awesome. I mean, she brings
Yeah.
on awesome people to host great rooms, and so like all the brains and geeks and nerdy folks will show up here, and it does create it's a nice environment where uh, all like minds kind of like come together.
Yeah.
So it might not be like thousands of people, but it is definitely the right set of people that kind of show up.
Cool.
Yeah, thank you for that great introduction, actually. And yeah, I think we can start. So, welcome everyone to Science Society, and a special welcome to Dr. Alfred Hero. And before we start, let me give you a little bit of introduction so you get to know him a little bit. Um, Dr. Alfred Hero, he's the John H. Holland Distinguished University Professor of Electrical Engineering and Computer Science, and the R. Jameson and Betty Williams Professor of Engineering at the University of Michigan Ann Arbor. And he is a fellow of the Institute of Electrical and Electronical Engineers and the Society for Industrial and Applied Mathematics. and he's a recipient of the Fourier Award in Signal Processing from the IEEE, and he's a section editor of the CM Journal of Mathematics, of Data Science, and senior editor of the IEEE Journal, and selected topics on in signal processing. And yeah, so we are very honored to have you here today. And before we start, we like to usually ask like a couple of interview questions before we dive into your project and your research.
Cool.
Uh, yeah, so how did you come about to choose science as your life or career? Was it something you always wanted to do? Was there a teacher, maybe a class you took, a book you read? What brought you to this life?
Yeah, it's a very good question. I was born. I'm one of the baby boomer generation, sort of on the later end of it. Um, Born in the 50s, grew up with science fiction, which certainly was a, a titillating introduction to science as it affects society and affects humankind. Jules Verne and H.G. Wells and Stanley Kubrick, of course, later on in 2001. Of course, it was also a very turbulent time with the Bay of Pigs, the Cuban Missile Crisis, JFK, MLK, RFK, all those assassinations, and then Apollo 11. So it was, a period of lots of turbulence. Um, but with my interests that were effectively coming from a combination of the books that I read, the science fiction primarily, and the realization that society was in turbulence. Um, it was a desire to do something in terms of a career that would both create new scientific solutions to problems that uh, would affect society. And so, I went to high school and was really taken with physics. If there was one teacher, it would have been my high school physics teacher in my ninth grade, who really got me hooked on experimental science. And then, of course, I got more interested in math, mathematical and computational aspects of science as I sort of moved into college, and then came into contact with scientists from all stripes, not just computational scientists, but bench scientists, experimental scientists, and that really motivated me to do a doctorate in this in the area that I've perceived as being the most central to science in general, and that is the computational aspects, the computational underpinnings, which affect everything from astro sciences, astrophysics, solar physics, to health, to biology, material science, and beyond. All areas that I've been very active in during my career in trying to contribute computational methods to enhance and accelerate scientific discovery. So that's sort of a
That's
description. Yeah.
that's fascinating. It's always so interesting to hear how the path led to to science.
Mm.
I think it's and how did you then, from discovering that this was kind of something you would you find this really interesting, how did you come to um, doing research in this specific area? And and is there a specific story to doing research in this microbiome? Because you're an engineer, so it's really
Yeah.
interesting how this came about. Really.
It's funny, yeah, because in the beginning, my first real encounter with microbiome research was about 7 or 8 years ago, when there was a initiative here at the University of Michigan called the Blue Sky Initiative, where people were encouraged to think out of the box about things that would be especially propitious to study in consort with multidisciplinary teams, and that could have really transformative impact. And so a colleague of mine in engineering, in chemical engineering and mechanical engineering, at that time was thinking about how to make antibiotics without the biotic part. So using nanoparticles instead of antibiotics to form microfilms that are biofilms, I should say, of microorganisms that could replace antibiotics. So, in a sense, my entry to microbiomes was through synthetic biology as opposed to true microbiomes that exist in nature. And then that led to, of course, you get one thread and that leads to another, and led to collaborations with the University of Wisconsin that on the actual, in this case, gut microbiome. I also have another collaboration there with another scientist, Joe Handelsman, on the plant microbiome, the rhizosphere. So it's become a major component of my activities because it combines sort of all those aspects that a scientist loves, and that is an enormous impact if you can make a difference and discover the inner workings and mechanisms of microbiome interactions with the human host or with any other host, and the computational challenges that lie underneath, which I'll talk about in more detail as I get into my talk.
Well, that is really interesting, and I'm so glad you were open to kind of venture out of your I don't know if it's really out of your field, but yeah, to venture into these topics that maybe in the beginning when you started your career you didn't think into going into a more biological complex
It's
system.
You're true. It's absolutely true. I did not have more than maybe one class in biology my entire university career. I think I had one class in biology. That's right. But coming from the computational side, I could not do a wet lab experiment. I'll admit that right out. Um, Ophelia Venturelli, my co-author on this paper, does all of the experimental work. But the computational aspects have become more and more important to scientific discovery, in particular in very highly complex systems like, these hundreds or thousands of interacting microbes in a gut microbiome that produce different types of functions, metabolites, volume functions, transformative functions that are extremely complex in terms of the composition, the types of bacteria that are present in the microbiome, and that's what we set out to find in this project.
Yeah, that's yeah, I still think it's really wonderful that you ventured out there, and based on research we have so far, which is not perfect, it's also the perfect way of staying healthy and young.
Mm-hmm.
Because being a rookie at something and keep learning, I think it's kind of one of the key
Yeah.
components of staying young. So
And
thank you so much for sharing that story. And yeah, you're welcome to give your presentation. We'll ask questions in the end, unless you would need really clarification. We invite you to use the chat so we can address those, so we get the whole presentation covered in the time we have. And yeah, the stage is yours. Thank you.
great. Okay. Well, I think everyone probably has been able to open the PDF file that has the talk on it. I have a number of slides. I'm going to go fairly quickly through some of them. there's sort of something here for everybody. There's some high level motivation for both the AI part and the microbiome scientific endeavor that AI has come in to help clarify. And there's also some more deep dive types of things. Very little mathematics here. There might be one or two mathematical equations max, but we'll see how it goes. So if you go to the first title slide is basically just a copy and paste from the paper that just appeared last June on recurrent neural networks for microbiome dynamics discovery. And I'm one of many authors. We're 6 authors. 3 of them are computational scientists. 3 of them are experimental scientists. So this is one of these examples of how putting computation in tandem, in concert with experimentalists, can really accelerate the process of discovery and design, in particular microbiome design, which this is targeting. Um, so the second slide, I'm going to just give you some takeaways. Now, you may not understand all these takeaways right now if you don't know anything about the microbiome, but or machine learning. But I just give these takeaways at the beginning to sort of provide some high-level context, and then we'll slide down into some of the more specifics after I finish talking about the main points here that come out of this paper. So number one is that this computational model, which we call machine learning, which is of course an umbrella term for lots and lots of things, probably one of the least well-defined terms in computational science, because it covers almost any kind of mathematical model that you might apply to data in order to predict responses that occur under conditions under which the data has been collected. So computational modeling, machine learning, accelerating discovery is really the whole bailiwick of our activity. And in this case, we use this in microbial communities with experimental data that's been collected by our Wisconsin colleagues to learn relations between the species that are in these microbial communities. In particular, this is a synthetic gut microbial community. Synthetic, I don't mean synthetic in terms of not biological. These are actually biological communities, but 25 different species out of the over 1,000 species that we know naturally inhabit the gut microbiome. So we look for composition species, the species that are in each one of these communities, if you like, growth over time. Where do they end up in terms of stable relative abundance, as it's called? Abundance meaning how many microbes of one particular species are there in the community, and how does that fluctuate over time? And then metabolic function, which I'll talk about several of the types of functions that these microbiomes will cause in a minute. So to give you an idea of the complexity of this problem of trying to learn these relations, there are, with 25 species, there are 33 million communities, 33 million possible combinations of 25 species, from communities of 1 all the way to communities of all 25. Well, one community of all 25, right? And so we can't perform that many experiments to discover how these communities affect the growth of the different species in the communities, or the metabolic function that they induce. So we're reduced to only looking at a handful of experiments, 413 to be precise. And we're able to basically extract from a single time point of what we call a full metabolic yield dynamical profile that enables us to identify particular bacteria in a microbiome that specialize in growing the microbiome, that is, growing the volume of cells, bacterial cells, or function, growing the function, the metabolic expression, if you like, that leads to being able to digest foods or maintain gut health or beyond. And so we accomplish this in point 3 by really embedding machine learning into the scientific discovery process. So this is not a case where the computational scientists worked on a model or pulled a model out of a TensorFlow um, uh, that then was thrown to the data, and then we communicated by throwing the results back to the scientists. We worked in tandem, meeting virtually every week to really embed machine learning into the process, and I'll discuss how that works. It's a 2-stage process that we implement in the paper. You perform a handful of experiments. You seed the predictor with 300 and some odd experiments. And then you use the outcomes of that predictor for that very few experiments to try to predict the 33 million community behaviors. And that's where machine learning is so useful, because it can generalize quite effectively from small amounts of data to the larger scope, if you like, of possible complexities that might exist in this case, the communities. Okay, so that's I'm going to go to the next slide here, which is just showing this this scientific discovery cycle, sometimes called the Kuhn cycle, that that characterizes how scientists do scientific research. You do experiments. You then perhaps infer from the experiments a potential model for why the response happens when you have particular conditions in your experimental apparatus. You then go back and generate more data, learning from that model how to predict what the best next experiment would be to explore the landscape of possible experimental conditions to produce responses of interest. And so putting machine learning into this loop is what our research is all about. And so automating parts of this, either the learning part to inform, that is model building, or the experimental part, using the model to most successfully designate potential experiments that will allow the model to actually improve itself over time. And so you have this cycle where we can incorporate machine learning at various points. And so I'm going to go now to the microbiome on slide 4 for the gut microbiome shown with the human and the digestive system in the middle of the slide. So a microbiome is really just a microbial community, right? It's several types of microbes that are denoted at left of this gray box that all interact together. Some of them interact in a inhibitory fashion to inhibit other species of microbes from expressing a particular type of compound, metabolite. Or it may enhance, which is that arrow, green arrow is denoting. And so these metabolites are chemicals denoted on the right of that gray box that then can enhance the ability of the host, where the microbiome is located, to function, or it can prevent disease, enhance human performance, and also produce valuable compounds. So, for example, you've probably all heard of with the different oil slicks that have been mitigated by applying bacterial films, bacterial treatments to mitigate negative environmental impact. And actually, these bacteria eat the oil slicks away, right? And that's a microbiome in terms of ingestion and production of particular compounds that dissolve the oil slick. So production of valuable compounds is a very important aspect of microbiome design. But also, we're now realizing that that one can design human microbiome communities by introducing, for example, microbiome supplements, if you like, probiotics, as they're sometimes called, yogurts or other types of supplements into the gut microbiome to enhance its function or to mitigate against disease. And so going to slide 5 gives us an example for a particular set of what are called metabolites, these chemicals that are produced by these interactions, complex interactions between microbes in the microbiome, lactate, succinate, acetate, butyrate. These are 4 different products of microbiomes that are produced over time. So they fluctuate over time according to both the circadian rhythms and the relative health of an individual. And they have different functions, if you like. You can see in blue here, written quite small, so maybe you want to magnify your screen. A lactate is known to be a athletic performance influencer, and succinate is highly associated with diabetes and glucose homeostasis, and acetate for different syndromes if it's out of whack. If the acetate production is either too high or too low, you can have incipient problems, dysfunctions. Butyrate has lots of different functions too. So each one of these has different functions. They're produced in different quantities by the different types of communities, microbial communities, microbiomes that might exist, the microbes that coexist in the gut. And so going on to slide 6, this sort of gives you a summary of the gut microbiome in terms of its function to perform both chemical transformations and physical transformations. These gut microbiomes grow. The number of microbes grows. They reproduce and produce different abundances, filling volume, creating a mechanical type of transformation in the space. So bloating, for example, that we've all seen when we eat too much, right, is caused by that kind of compositional modulation, in addition to gas, which is a particular type of metabolite production. The role, of course, is, as I just say, maintaining human health. It's a dynamic community, as I tried to emphasize. The microbial interactions, they really occur as mediated over time due to various types of conditions. Whether you've eaten recently, whether you're exercising, whether you are sleeping or awake. And most importantly for the computational part of this is now we're starting to talk about modeling a little bit. The interactions between microbes can be either pairwise, or they can be what we call higher order. In other words, that you may need to account for how groups of microbes co-interact with each other, that in order to understand how these microbiomes produce different types of function. When we talk about function, we're talking, again, composition, abundance of species, or metabolite ingestion, metabolic yield, as we call it. And so that brings to the fore microbiome engineering, which we want to design the gut microbiomes to have a particular multifunctional profile. Multifunctional meaning a particular profile of metabolite production, right? Or a particular profile of growth. Um, and that then will allow us to develop different types of microbiome treatments, microbiome supplements, synthetically uh, in the lab, but also using models which will allow us to understand how different types of communities, that is, combinations of species in the microbiome, um, will affect both this microbial abundance, which is in orange here. You can see that the axes here are time on the horizontal, and xi is the abundance. That's a number, an integer, right? That tells you how many microbes of a particular species exist. So the white curve is one species, the black curve at the close to the bottom is another species, and so forth. And then metabolite concentration. That's the metabolite or metabolic function. Concentration is denoted by C, and again, that evolves over time. And so the main challenge, of course, to understand how any particular community, any particular combination of these microbes in these 25 species that I'm showing at the left that have been now put into an experimental multi-cell incubator to basically form a bioreactor to do experiments of different combinations of these communities. You have to explore all the communities if you want to have a complete, comprehensive understanding of the microbial abundance and metabolite concentration that would occur for any of the 2 to the 25 minus 1 exponential number of distinct communities, right? That you might make as a microbiome designer, right? So it's obviously impractical to experimentally test all of these uh, uh, possibilities. I'm going to slide 8 here and for these 25 species, there are 33 million distinct communities. Um, and so these are just some of just shows you some of the breakdown of 25 single species communities, about 4 million 11 species communities, 125 species community. So we have to somehow grapple with the problem that we're never going to be able to do experiments in all these communities, right? That's completely out of the realm of any reasonable experimental, even automated system. So an alternative to exploring this entire design space is machine learning, or building a predictor, right? And all this really requires is that we do a few experiments to learn more or less, that is, to some accuracy, how to map the measurement of the microbial community species composition, and maybe the initial conditions of how many species we started with before we grew them, how many species, how many species of each class we started with, and how that maps into a function, right? Growth over time, metabolic yield, and so forth. So you start with a small number of experiments to learn this predictor, this what we call input-output map in engineering. And that map is often called a machine learning algorithm, or a predictor, or a computational model, right? That brings you from a independent variable, microbial community, initial conditions, and species composition to the response, right? The function of the community. So the main challenge is, what do we use as a predictor, and how do we fit it to how do we fit that predictor to some subset of these microbial communities? It may be even a very small subset, like I was just alluding to a minute ago. And so in the paper, we adopt a what's called a recurrent neural net. So it's a neural net of architecture, which is model free, in a sense that there's no sort of underlying physical model that we are trying to emulate on this predictor. A few years, 1 year ago, there was a paper by our collaborator, Venturelli's lab, Ryan Clark, who was postdoc in her lab, um, on a model-based approach, which used a very intuitive a kind of, let me say, parallel between communities of bacteria and how they might grow and how might they might compete with each other to grow and to and to be killed in a similar way that a predator-prey model works for a pair of species, um, where there's a intrinsic um, trade-off between the food supply that that will allow lots of prey to roam, um, and the number of predators that will allow the predators to feast on those prey. And so these predator-prey models are given by a differential equation that I'm showing here on the left. I don't expect anyone to understand this equation. This is just to get across the idea that this is a mathematical model, right? It's a model that depends upon, in particular, what are called pairwise interactions. Xi is the abundance. So if you look at the function model, there's a multiplication of two of a pair of Xi, Xj. So there's no higher order terms in that model, right? So there are no higher order interactions in the different species that might be predator or prey relative to each other to reduce or enhance their populations. And so this was applied to a phyla of bacteria, 25 bacteria that are listed on the right, that are characterized by 4 phylum, Bacteroidetes, Proteobacteria, and Echinobacteria and Firmicutes, different types of bacteria, as a model for the most representative part of the microbiome in the gut. And so this paper derived some predictive results. There was a lot of deficiencies of the approach that led Ophelia to approach us and for us to get interested in this. Mainly the fact that the model was only pairwise dependent interaction, and it was also a model that was kind of grafted of a growth model and a function model were grafted together. There was something not so elegant about having to basically take two different models for the growth function and the metabolic yield function. So we came in to improve on this predictive model. And so this is so-called generalized Lotke-Volterra model, is the model that I just described. And it has lots of restrictions. I'm not going to go into these. Training the model is very heavy. You have to solve a nonlinear optimization problem. There's not easy tools for doing that, unlike in machine learning, where lots of tools have been developed recently to solve these types of problems involving backpropagation, as it's called, backpropagation optimization. So in this paper, we have a unified predictor model based on what are called recurrent neural networks. This is on topic 2 here of page 11. And it exploits higher order interactions. It predicts composition and function, that composition meaning abundance of species, their growth over time, and the function of metabolic production over time. And we can predict multiple time points for both metabolic function and composition, something that the previous model could only do for the composition. So we get much better accuracy. I'm going to now slide 12, where I'm just going to very briefly describe what recurrent neural networks are in terms of their history. And I'm not going to go into any detail on what the recurrent neural network is. That's for a different talk, probably for a different audience. But suffice it to say that recurrent neural networks are a type of machine learning uh, uh, process or procedure that's been around for a while. You can trace it back to the 80s. It's was developed primarily for speech applications, automated speech recognition, speaker identification, things like that involved temporal processes. Speech is, of course, temporal. Streams of text is temporal. Microbiome growth is temporal, right? So that's why we adopt these recurrent neural networks. Um, and you may be familiar with Google Voice or Google Allo. Those are two Google services for speech recognition and message suggestion and text processing that are based on recurrent neural networks, in particular on LSTMs. So at the bottom, you see Hawkwriter et al. LSTM. LSTM, going to the slide 13, stands for long short-term memory. And this is a module for the recurrent neural network. It implements a dynamical system, just like the Lotka-Vitero model implemented a dynamical system, a differential equation, but it doesn't explicitly specify the interaction type, right? Where it's pairwise, third order, fifth order, what have you. So it's much more flexible in being able to model complex interactions between between lots of microbes in high dimensional systems. So we're going to compare that to the predator-prey model, the GLEV model, and what we're going to find, and I'm going to very quickly go through these findings, is that the machine learning model, this LSTM, is as good as the predator-prey model, GLV, when there are only second order interactions. So it's able to perform as well in predicting what microbial communities that have not been seen in the training data, what kinds of abundance profiles, composition will occur in those if you started a community off at time zero and allowed it to grow. So generalization from a small amount of data, LSTM is as good as GLV when that data is synthetically generated from a model that only incorporates second order interactions into the generative mechanism. It's better than the predator-prey model when there are higher interactions. And so this picture comes out of this is on slide 14 from the 2022 paper, figure 1. And I'm not going to describe all these graphs here for those that are familiar with scatter plots and comparing predicted accuracy to relative to the true ground truth. You'll recognize these. But suffice it to say that what we did in this simulation was we took all species up to level 6 communities, right? That is communities that had 6 species or less. And then we generated the growth using basically the predator-prey model, right, for those less than or equal to 6 species. And then we applied our LSTM model and applied the GLV model to predict species that weren't, communities, I should say, that weren't in the training set, and composed of 11 and 19 species, respectively, right? So we have what are called in machine learning a training set of less than or equal to 6 species communities, and we have a test set based on communities that have lots of species, 11 species and 19 species, respectively. And so what these plots show is the predicted abundance on those large, complex communities from this model, this predictive model that only saw very small communities, right? Less than or equal to 6 species in each one of the communities. And if you have second order interactions in our simulation, you have comparing the top ground truth, the top GLV predictions to the bottom LSTM predictions, you have close to the diagonal line. The diagonal line, of course, is where you want to be. You want the predicted abundance, predicted species, relative species abundance to be close to the true abundance, right? And as you start to mix third order or higher order, you get GLV is on the rack, right? It's not able to do nearly as well. That line, that prediction line, is falling, right? So it's that diagonal that should be for an ideal predictor is falling, and we are now in a situation where the LSTM is maintaining its performance while GLV is suffering. Okay, so that's a simulation. This is just to show that the LSTM is doing what would be expected of a computational algorithm, a machine learning algorithm that doesn't have this second order interaction baked into a model, right? So the experiment that we did, I'm going to slide 15, was to take these 25 species and create 413 communities of the possible 33 million to then do these 413 experiments with 1.1 to 5 species and 11 to 19 species out of the 25 possible, the 2 to 25, excuse me, possible communities, and then apply the LSTM machine learning method with a feed-forward network for those who are interested to map the LSTM to particular outcomes, response variables, these butyrate, acetate, lactate, and succinate, or the growth curves, right, that are shown here in the left in orange and the metabolite curve in blue, right? So we trained this on these 413 communities. This LSTM was trained using a backpropagation algorithm that I'm not going to mention, with cross-validation to assure that it's not overfitting. And then we use this LSTM to predict the function of the remaining almost 33 million communities. And these predictions were used to inform a second validation experiment to learn what was, we were hoping to be the most diverse set of predicted communities, right? So with the 413 that we chose were kind of randomly selected. And then for the experiment 1, for experiment 2, we're going to target particular communities for experiment based on our desire to produce experiments that have communities with much more diverse microbial abundance and metabolite concentration profiles than what we saw under the random experiment. So I'm showing now on slide 16 the outcome of that experiment. What you see in the middle is this big square in the middle. That is what those gray dots are, the representing the 313 training samples and what they produced in terms of lactate and butyrate. So there's a pair, it's a point in the plane. What you see that's looks like a gray to black surface with some ellipsoidal shapes that are in the middle, 2 ellipsoidal shapes. Those are the predictions of the 33 million. So where you have very high, dense black, it means that there are a lot of communities in that at that point that produce butyrate and lactate at that particular level that is indicated by the horizontal and vertical axis, right? So we have 33 million communities that have now been predicted from 313 communities that they predicted the metabolite prediction profile. And here I'm just showing the endpoint of that profile that the predictor predicted at 48 hours, right? So what you see then is a landscape, right? That's what we were asking for. We want to discover a landscape of what the metabolic yield, the succinate, lactate, acetate, butyrate production is for any combination of these species, any one of these 33 million communities. And that's what we have now. Now, problematically, of course, it's only a predictor, and it's a predictor based on a very small number of samples, right? So what we're interested in is how good does that predictor perform for these points that we haven't seen yet, right? The ones that aren't marked by these gray dots. And so what we decided to do was do a second experiment with 95 different communities, where we chose those communities from these what we call corner cases. They're off on the side so that they have either very low butyrate production or low lactate production at the bottom, right? Or low butyrate at the left side, right, of this square. And we had then a few other communities that were chosen sort of in the bulk, as I call it, where we have just to validate in that dark region where there's lots of communities that tend to produce the same lactate butyrate combination. And then I'm showing, different pairwise slices through this four-dimensional space of these four uh, uh, different types of metabolites that are shown in green, purple, blue, and red insets. So what we did, as I just to reemphasize, we went in and we chose new experimental uh, planning communities. Right? How do you plan the experiment with these communities that are chosen from these corners to both validate the predictor, right, and also to update the model to come up with a better predictor, right, to incorporate those new communities into the model to boost its accuracy in regions where there's not a whole lot of evidence, right? In these corner communities, there weren't a lot of uh, uh, data from our initial experiment. So I'm just going to go to slide 17 very quickly to mention that a side benefit of these machine learning methods is their interpretability. And so what I show is that a method, a explainable AI method that we applied to the LSTM to elucidate what the most important species on the left in D of this figure, what are the most important species for predicting various of these metabolites, right? Butate, acetate, lactate, and succinate. And so what's in red are positive, what's in blue are negative relations, right? So a positive relation means that if you increase, for example, AC, which is a firmicute bacterium, then you tend to increase the amount of lactate that's produced. On the other hand, you tend to decrease because we have a red, big, thick red line going up to butyrate, connecting AC and butyrate. You tend to decrease butyrate, right, if you increase AC. So that's what that means. And so this machine learning algorithm has effectively enabled the biologists to identify what the most important species are for producing various types of metabolites, and how they might inhibit production, right, of certain metabolites. So if you want to produce butyrate, you probably don't want AC, right, to be in that community. So you want to pull it out and let some of the others contribute to butyrate, like DP and EL. What I'm showing on the right is the same picture, but for the abundance, for predicting the abundance that occurs over time. And we're looking basically at what are those pairs, those interactions between different metabolites that tend to increase abundance versus decrease abundance, right? And so again, red and blue correspond to increasing versus decreasing abundance when that interaction is present. And so again, this gives us sort of an interpretable uh, um, uh, mechanism for explaining which variables are the most important in both growth and, on the right, and metabolism on the left. And one of the most exciting findings that we came up with is this picture, is that you can notice that out of the 4 different phyla of bacteria, really only 3 of them are dominating the metabolite production: firmicutes, actinobacteria, and proteobacteria. The bacteroides are not really making a big entrance here. They're not. They're not. They don't have very much influence. Um, PC is a bacteroides, and it doesn't have much influence on the left. It's only influencing one succinate, and that's being taken care of by others that are in the actinobacteria and firmicute. On the other hand, for abundance, bacteroides are the ones that are governing the growth. So their bacteroides seem to be associated with the growth dynamics of the microbiome, whereas these other types of bacteria are most specialized, if you like, in producing particular types of chemicals, right? These chemicals that aid in digestion and other functions. So I'm running out of time, and I'm going to skip ahead to experiment 2, where I mentioned that we tested 95 representative sets of communities predicted by experiment 1. And the punchline with this is that we observed a diversity of these metabolite concentrations when we performed this experiment, and that's on slide 18. You can see that there's a very diverse set of profiles of these 4 different metabolites, and that was the purpose, was to explore the landscape and then validate that landscape and then improve the model. And that's what slide 19 is all about. Is we now incorporated these 95 additional experiments into the original experiments, and then retrained the LSTM machine learning algorithm, resulting at the bottom left with a cross-validated prediction accuracy on a leave one out basis that illustrated that the LSTM was performing very well, and also elucidating again the differing roles of these various on the right of these various species in producing these different butyrate, acetate, lactate, succinate metabolites. Okay, so I'm going to my conclusions. Slide 20. We have established that embedded machine learning can be used to accelerate scientific discovery, reducing the number of experiments. If someone had tried to perform just a randomly selected 413 experiments, you wouldn't have discovered those corner cases because they're way out in the tails, right? The probability you would have discovered those would be infinitesimally small. But by using this 2-stage process, we discovered those through the predictor, and then we're able to very quickly zero in on them, I should say, in the second experiment. We have explainable AI that helps us interpret the model, and useful design principles have emerged, and I just mentioned those. The drivers of metabolic yield versus the drivers of growth dynamics are 2 different classes of micro bacteria, right? So future directions, we're looking to do a validation on in vivo. This is all done on a synthetic microbiome, right? So 25. Can we validate this on actual gut microbiome? Maybe a mouse model to start out with is what Ophelia is suggesting. We have fixed the environment and the growth media in these experiments. What's the effect of those 2 things on growth and metabolic function? We don't know. We'd like to do additional experiments with machine learning embedded to find out what the optimal environments are and growth media for attaining particular outcomes, functions. And then we're looking also, as I mentioned in the beginning, applications to different types of microbiomes like plants and antibiotics. And then the final thing I should say, which is more on a methodology and sort of scientific exploration direction, is we're really looking for a deeper, beyond two-stage embedding of machine learning, so it becomes intrinsically part of the design cycle, incorporated into an automated research workflow system. And I'll just, by way of explanation, go to the last slide in the talk, which is just basically a pointer to a report that I was participating member on, chaired by my colleague Dan Atkins at the University of Michigan, and reporting on a study that the United States National Academies did on automating the research protocol and procedures using automated research workflows for accelerated discovery. So with that, I'm going to stop and see if there's additional discussion.
Thank you so much for this wonderful talk, for explaining this, especially the machine learning part to us really well. So I really appreciate that. And yeah, if anyone has questions, please flash your microphones, um, and yeah, and we'll go from there. So Dennis, Joyce, you flashed your microphone, and Eric, and I guess Mayor and Dr. Shaw. So yeah, let's just go in PTR maybe. Thank you.
That was a fascinating talk. I definitely have more questions than we have time for. I'm going to ask a really sort of basic one. So there were 413 out of the
3 million.
33 million possible. So during experiment one, how were how was that initial set of experiments pulled? Like, was it representative of like, how did you select them?
Yeah, it's a great question. So we had the benefit, we all stand on shoulders of giants, right? So we had the benefit of over 2 decades of research into the gut microbiome that had specified, that either in single shot types of experiments or in multiple shot, like what Ryan Clark, the postdoc in Ophelia Venturelli's lab, had performed. So these species were selected so that they would highly represent what we knew, because they were skeptical at first. I think I think if you asked Ophelia, she would say that she wasn't completely buying into this idea that you abandon this tried and true model in ecology and predator prey, to a model that is not based on a mathematical mechanism, right? So she wanted to make sure that we chose a set of communities that were reasonably well understood, and that then of course there was a set of other communities. I forget the exact proportion that were chosen sort of at random, right? Sort of as just a way to validate how well we might do in using these machine learning methods. Does that answer your question?
Yes, absolutely. I definitely appreciate it. I want to know so much more about how the machine learning goes, but I want to leave some time for others. So, Dr. Shaw, to you.
Yeah, thank you so much. That was absolutely fascinating work. My question is about use of your research for the whatever that you know as a fecal oral transplant, because you
Oh
know
yeah,
that we have a IBD
yeah,
condition, and I was just wondering maybe we can use your research and see that what is your opinion.
yeah. Well, I think these fecal transplants are they're fascinating, and they seem to have had not just for IBD, but in other domains, have determined everything from the cognitive states of in mouse experiments, to different types of performance in, again, primarily mouse experiments. And of course, there have been human experiments with fecal transplants that have had very good initial results. I think that, you know, the implication here is that a transplant that's not necessarily fecal, that may be from perhaps even a a say greenhouse, to use an analogy, where you use bioreactors to grow different species, create the communities synthetically. You don't transplant them from another, you know, healthy human being to try and solve the dysfunction of a IBD patient. Um, you basically create them in the lab, and then you add them through a capsule, right, or a treatment. You swallow you know, a refrigerated capsule, and that would be composed of synthetically designed but biologically, you know, living bacteria. And this research would help to uh, sort of tease out which types of communities would be the most effective in you know, treating certain types of disease or dysfunction based on our understanding, which I must admit is very limited right now. If you ask a gastroenterologist, certainly they won't be able to tell you that you know, it's a combination, this particular combination of lactate and succinate that causes fecal transplant recipients to improve in their in their prognosis and symptoms. But once we have that understanding, we'll have an ability to link the particular response variable that we've used, which is this abundance and metabolite concentration, um, to you know, the particular type uh, of compounds, chemical compounds that are required in order to have a lasting uh, uh, treatment effect. So, yeah, I'm not a clinician, so I can't really answer any deeper than that. But that's, I think, a very exciting area for this type of research to have impact.
Also, for the species that you just collected, did you use the whole GI system? For example, duodenum that we have, for example, 100 million per gram microorganism there, or is just specifically from colon?
Right. These 25 species were selected by my collaborator. So I can't really speak too much to why she selected this particular set of 4 phyla of 25 species, but she was certainly targeting the gut microbiome. I'm not sure whether it was the colon or the you know, the stomach, or what part of that microbiome she was trying to target. I think she likely choosing these on the basis also of experimental feasibility, right? Um, Bacteria that are fairly easy to grow and maintain and resilient to environmental conditions and so forth, just to sort of get started. But she'd do a much better job at describing the selection justification for these particular 25 bacteria. But a great
Thank
question.
you. Thank you so much, Alfred.
Yes, you're welcome.
All right. So that was awesome. I got some questions. I
Sure.
have more of let me kind of explain. Can this thing progress to the following, right? So what I kind of see here is you can go in there and basically start to, you got 30, would you say 33 million communities? That's not species, but 33 million different communities.
Different
That's what
communities, right? Yeah,
holy cow.
25 species. Now, if you go to the thousands of species that we were just talking about,
Yeah.
there's going to be a heck of a lot more than 33 million.
Yeah. This is wild. So you can basically go in and do like very personalized medicine, right? Where you go in, once you keep expanding this model, get more and more data, the predictions are getting better and better, right? Instead of
Right.
doing like a fecal transplant from somebody that's healthy, but you're getting a whole bunch of other unknowns in there too. But you might want to just get a very specific type of community growing with the bacteria. That's where the capsules can come in come in and inject and get that community going. But I can see this becoming more better and personalized with this model, because then you could probably like pull out data, basically pull out a specimen from an individual, put them
Yeah.
into this model, and then now you can get very accurate understanding of what you need to deliver in that capsule down into the microbiome to open up and basically start creating some type of new community of some sort that's specific to that person, according to that percentage of what he needs. Is that kind of like accurate?
That's a great suggestion. Yeah, and the application of personalized medicine would be one instance of it, where you basically take a sample from a patient, right? That you then grow in the lab. You look at the metabolic functions of that microbiome in conditions that emulate that person's gut, and then you try to identify from the landscape of all these other communities that we've predicted with other people with similar types of environments in their gut, what would be the supplement, right? That would supplement bacteria that would put that particular person back into the healthy phenotype. Um, and so that is probably way in the future, but it's I mean, these experiments are expensive right now. It's a process to, when you look at metabolite production, it's not instantaneous, and it varies as a function of the circadian cycles and digestive cycles. So you probably have to take samples over a period of time during the day. Or maybe even during a month, right, to account for the different types of lifestyle variations that the individual might have that would cause changes to the microbiome. You could then build a microbiome temporal profile, if you like, that's waxes and wanes in certain dimensions due to these various factors: environmental factors, dietary factors, exercise factors, and that would give you a much fuller picture of that individual. And maybe that one thing we haven't discussed at all, we really even haven't explored, is resilience, right? So some of the what we have, this microbiome that is synthetic, in vitro, I should say, microbiome. It's in static conditions. Um, but if you start to look at resilience, then you're asking, if those conditions start to change, maybe these results that are being predicted by machine learning, um, those only apply for particular conditions. And so what you'd want to do is you'd want to use maybe some sort of uh, what's called adversarial machine learning to account for the fact that you want robustness of the predictions to these types of environmental variations, right? And that might give us completely different set of bacterial membership in these communities that produce these various types of metabolites. But it's a great suggestion, very, very interesting idea.
Yeah, and lastly, I think like because it's everybody's microbiome is different, right? So and then you're dealing with living microbes. So I mean, I could foresee like once you got this model down, it would be very beneficial to almost like go show up somewhere and go in, and then they take a sample and they come back and say, okay, well you need this and this and this, and they make you a concoction of like different
Right.
capsules and things to take that are specific to you. That then you take home, and then you basically utilize, and then you go back and forth, and basically get your microbiome fixed up in a very laser focused way.
Yeah. Right. Right.
It's like in a
And
few years, it'll be microbiome synthesis as a service. We'll see that pitch deck,
Exactly.
I'm sure.
exactly.
Exactly.
Right. Yeah. No, there's a lot. And then there's sort of the implication that there's another step here, right? What we'd like to be able to do is, once they've gotten these capsules and we've figured out what they need, we'd like to figure out how they would naturally be able to supplement, right? So they're not dependent on the capsules anymore. And that's much, in terms of a business model, that's a much harder proposition, right? Clearly. But I think in terms of social benefit, it's unless you go that entire length of trying to develop a in vitro solution, and then leading to a natural solution where the person can adjust their diet, perhaps, or use some other mechanisms, including things like exercise or change of lifestyle, to try and correct their microbiomes. There's so much that's not understood, and microbiome, of course, it may not be the root cause of the malaise or disease that is causing the sort of dysfunction of these communities. So trying to go to the root cause is I think, an important second step, if you like.
Sure, sure.
Yeah.
Yeah, I'll go ahead and I think Ayo, you're next, buddy.
Yeah, thanks. So my question is about the interference terms, and I guess also similar to what
Heck,
Dennis was saying
heck,
with the select.
can I just put a hand up? I think we've skipped me, but I'm happy to go after Io.
Oh, maybe pull to refresh. My
Yeah,
bad. I guess.
Yeah,
yeah. I think
yeah.
Russ was first, and then Eric, and then
Oh,
Joyce,
I'm
and then
sorry. On my screen, no,
um,
on my screen it had
yeah.
Ayo first. I
You
apologize.
just got to
And
refresh.
in between, I wanted to check with Al how much time you have left because it's
So I have till 3:30. Yeah.
okay.
So
Okay, so
yeah,
everyone
that's
one
another 15
question,
minutes or so.
then we should
I'll
get to everyone.
Yeah.
Thank you.
I'll be exceedingly brief. First of all, really eye-opening and presented so beautifully. Like I always admire the capacity to metabolize dense information and then feed it to others in a way that they can climb on. And so, thank you for that.
Beautiful.
I'm thinking about sort of, let's say the microbiome has that level of specific effect on, the human and everything else, really.
Mm-hmm.
I'm thinking about some implications of that. One being, if we set about altering microbiome by person or community with intention about, well-being and, let's say, best possibility for species, we inevitably will be changing our relationship and the microbiome's relationship to everything else. Sort
Mm-hmm.
of a profound environmental impact, butterfly wing style.
Uh-huh.
So I think that's the one comment I'll make, and I'm interested in your thoughts about that.
That's a great comment, and it's it's certainly, nature has evolved. The our species and others, all of life on this planet, to have a homeostasis of uh, microbiome health, right? And if we try to shift that to a different operating point, maybe you could call it a local, a saddle point, uh, maybe a local minimum, but probably a saddle point. Then it opens up an opportunity for everything from what we know so well in the past couple of years, viruses or bacterial infections, to take advantage of the fact that we as humans have embarked upon, a blinking of an eye, uh, second of our in the era of multiple epochs of natural selection and natural development of the microbiome. We've chosen to shift it without realizing what kinds of vulnerabilities we are opening up to in making this shift. So I agree completely with you on that philosophical point, and uh, I think that this whole area deserves a much deeper discussion with people in the room who are the big thinkers on these what you call butterfly effects that can be catastrophic for the species or for life on Earth in general.
Or amazing if the
Or
same
amazing. Yes.
intention is applied
Yes.
in altering the microbiome in favor of, let's say, addressing the climate crisis.
Yes. Right. Right. And that's another aspect. Right. But what do we open in terms of vulnerabilities if we do that? That's the question. I mean, we can address the climate crisis symptomatically, to use a clinical analogy. But if we're not addressing the root cause by reducing fluorohydrocarbons and other types of production that increases our exposure to climate induced changes, then a microbiome difference may be just a drop in the bucket, right? That doesn't propagate too far. But fascinating ideas, and I think raising a lot of issues that go way beyond sort of the bench science that we're talking about here.
Yeah, hi. My question is on page 10. You have on slide 10 or whatever you want to call it.
Yeah.
Function model regression complementarity effects. Does that include positive and negative effects, or is that only? Because I was just curious, because like we're looking at kind of the viral version of what you're doing, and we're trying to see interference terms. And so it's really hard if unless you already know kind of the molecular situations that are going on. So I'm curious as to how you made choices in that part of the model. Thanks.
Yeah. So first of all, this isn't my work. This is the work of Ryan and Ophelia Venturelli. We sort of built on this work as sort of the launching point. This model is based on this predator-prey model, and you're undoubtedly familiar with it from your prior work on the growth model, the GLV, generalized Lotka-Volterra model, where there's a this IJ selection effects and basal growth basically control respectively the the trade-off between growth of the prey species in absence of the interaction with the predator, and growth of the predator species in absence of interaction with the prey. And when you put them together, that effectively describes AIJ. For the function model, the regression model, this is a regression that makes an assumption, and I think I'm a little biased here because I'm arguing here that we should not be tied too closely to a pairwise interaction model. One could argue perhaps that the pairwise interaction model might be justifiable from a predator-prey perspective, and maybe with only two agents, two bacteria in this case. But I think for predicting function, that's a much more complicated process. And
Yeah. So
so I'm
we were
not
looking at kind of the interference terms and saying, okay, we're going to assume n terms of interference, not just pairwise interference, and that got
okay.
and very quickly. And then we tried to think of ways of reducing the model so there was some kind of similarity in chemical bonds or approximations that you could do kind of to coarse grain it. But yeah, thank you. And sorry for not realizing the question kind of was slightly not related to your work directly. But in terms of that kind of decision making, like I guess just established databases provide the most informative
Yeah.
view. Yeah. Okay. Yeah.
I think so. Yeah, and I think if Ophelia comes on at a later time, which I hope she does, she actually wanted to be here too, but she's in Europe right now. Then come back, and I'm sure you'll get a lot more information about this model.
Will do. Thank you very much.
So I think maybe I'm next. I was going to say I did ecosystem modeling research quite a few decades ago at Oak Ridge National Laboratory, and I can say that I appreciate the difficulty of what you're doing, and it's
Mm.
really great. And a lot of advances have been made since then. And I was just going to relate a little bit more to a topic you already mentioned a little bit, but maybe you want to say a little bit more specifics about it. And that is about the role of diet and studies. There are various types of studies, some that establish what they consider to be an anti-inflammatory diet, and they even have an index, and then studies associating a westernized diet with various autoimmune and inflammatory diseases. And I put a link in the chat, and the studies by Chiba, epidemiology and interventions with plant-based diets in IBD. And I'll put a link in there for that too. Anyway, any
Interesting.
thoughts?
Yeah, I think that's a great question that relates sort of back to this this main issue of what can you do to perturb the microbiome that you have to something that's better, right? And that is more, both uh, gives you a more resilient ability to maintain, digestive health, but also one that is long lasting, right? And that's where diet comes in. I think that what you want to do is you want to get a sea change of the interactions that are possible and those that are not possible, right? Because some interactions can wreak havoc um, in terms of as we were showing in our results of inhibiting certain metabolite production, right? So if we could sort of break down in the sense of what kinds uh, of bacterial or microbial more generally communities would work best to accommodate, let's say, a dietary regime which is rich in lactate, right, already. So yogurts and dairy products, um, or for someone who's lactate intolerant, um, does it have something to do with the microbiome? Is it that the microbiome is maybe deficient in the bacteria that are effectively aiding the ingestion or digestion of lactate, um, so with plants, it's a similar type of story, right? Are there bacteria that are especially um, uh, well suited towards a dietary regimen where they, can pull the maximal nutritive value out of different types of diets that some of which may be deficient, right? Or some of which may be overabundant in certain types of components. I mean, clearly, it's been seen that in the research that fibers and uh, probiotics have a uh, a positive effect, if you like, for at least the evidence is that it has had a positive effect on digestive health. How that is mediated precisely by the microbiome, that's where we could start to look at different types of research. I mean, if we could synthesize in vitro a full digestive system where you can introduce different types uh, of food groups and environmental conditions and so forth, that would be an interesting follow-on experiment, with the caveat, of course, that it's also well known that that there are connections between the microbiome and the brain, right? The central nervous system. Serotonin I believe is at least there's some of the partially produced by the gut microbiome, right? Which, of course, serotonin is what allows the neurotransmitter function to uh, to, to thrive. And I think that having an in vitro system, there's always a danger, right, uh, of not treating the holistic whole of the host that for which the microbiome is so integral um, in terms of maintaining the host health.
Yeah, thanks. And then you add another layer of complexity about individual inflammatory allergy reactions to
Yeah,
particular things. That is a whole nother complexity level.
exactly. Yeah, autoimmune diseases, right? And yeah, that's just just takes it in a completely different dimension.
Yeah, thank you. We have 2 minutes left, so I'm not sure if we get to everyone that's still on the stage, but maybe Einar, did you want to ask a question?
Yeah, hi. I'll make it quick. So when it comes to this AI that you use, uh, is there any regulations here when it comes to difference in the microbiome when actually will transfer toward the spine and what will be further on processed by the human mind and whatnot? And as my hair was into also when it comes to capsules.
Mhm.
Does it say anything about the adaptation level or the time that we actually need to just like switch in a snap in towards capsules? Just like to me, it seems like this will take probably many years for a body to adapt to. Yeah, thank you.
Yeah, that's again a really great question because effectively these AI techniques are limited by the training data that you provide, and they if you don't provide at least somewhat representative training data, unless you supplement with mechanistic, biological, physiological, um, chemical models, you're not going to be able to predict very well, right? AI, from a mathematical viewpoint, is nothing more than a an interpolation, as it's called, right? You can interpolate between points, and you can extrapolate to a certain point between points, but it's just a big, complex interpolator. And so, if you don't have enough data to support that interpolation, in the right parts of the data space, then you're not going to be able to say very much about that part of the data space, right? Because it may not be well interpolated. So, in terms of doing the looking at things like interventions, and that's what these capsules are. That's a whole nother area that we don't even address. What point in the cycle you saw those metabolic and growth curves that I showed for each one of the species, and these growth curves, there may be some most propitious time to give that capsule, right? If you give it at that particular time, then the capsule will have the best effect. It will integrate the most rapidly and the most effectively with the rest of the microbiome as it exists. If you give it at the wrong time, maybe it just passes through, right? Um, so uh, it, again, these are really good questions that go far beyond what we've just sort of scratched the surface with these experiments. So I'll entertain one more question, and then I'm going to have to go.
Great, thank you. Go ahead, Rebecca.
So I don't know if this will apply to the talk. I missed the talk, unfortunately. I'm curious if the same repetitive movement of the body, whatever the movement is, affects microbiome.
Yeah, that's a question that goes beyond certainly my experience or expertise. Um, exercise has been linked to microbiome health. How, so is exercise a repetitive movement type of activity? Well, maybe, right? So when you say repetitive movement, are you referring to something different from exercise?
Yeah, I'm referring to any repetitive movement. So if it's like the slumping of the shoulders, or if it's
Hmm.
like a stop sign with your hand, like a boundary, or
Yeah,
a squat. If there's not time for a further conversation about it, but it will answer a question I've had.
yeah. I do not, first of all, whatever answer I give, you should take with a grain of salt because I'm not a specialist in microbiome physiology. Um, but I, I would, I would think that any degree of physical activity that can, restore uh, both sort of uh, a, a chemical and a mechanical environment for the microbiome to thrive would be positive. But it's just so linked with everything else. I mean, people. I mean, we've all had experience. If you exercise regularly, you have better digestive passage, right? Um, you have better appetite, and microbiome certainly is associated with appetite. That's one of the main functions of microbiomes is to regulate appetite in a healthy individual. So, I think it would be extrapolation to say that other types of repetitive activities would have uh, positive or negative effect on the microbiome. But certainly, exercise has been established as having a measurable effect on microbiome health and host health.
Great, thank you so much.
Yep, you're welcome.
Well, thank you
Good.
so much, Al. I know Armish had questions about the
I'll be very
machine
quick.
learning part,
Okay.
but
All right, very quick. Yeah.
So, thanks a lot, Alfred. I missed the initial part, but from the paper, I understand it's a time series modeling. So, I
Right.
just want to know, so the combination, the whole permutation and combination of the species group which you made, if you put in a GLV model versus if you put in a LSTM model, what's the T there? What's the time difference over which this gets observed? That's
Oh,
one thing.
yeah.
And after that model, can it also extrapolate handsomely to the time for which it hasn't seen in the raw input data?
Very good question. So we do know, I'll answer the last part of your question first. We do know that that it's very good at interpolation. So when we trained on our first experiment, we trained only on an endpoint metabolic production. So we trained at 48 hours. We had data from metabolism. Um, for the other 8-hour intervals, we had data on abundance, that is, on species relative volume, right, within the microbiome. But we had no metabolic information beyond that endpoint. And yet, the LSTM time series model was able to induce what the metabolic profile was. Now, most likely that was because of the fact that we had time series on the abundance of the relative abundance of different bacteria, and that's related. That was sufficiently related, right? The waxing and waning and relative uh, number of species translates into metabolic production, right? Also a waxing and waning of metabolic. So we thought that's probably the answer there. Now, in terms of the time experimentally, the time that you can reasonably do an experiment, the turnaround time, if you like, for one experiment is about 8 to 16 hours. And the reason is that the medium actually becomes corrupted after a period of time. The medium changes, and so the bacteria ceases to grow or dies. So you have to basically refresh the medium, which the experimentalists did at 16-hour intervals. And so that corresponded to the challenge that we had is that, while certainly microbiome dynamics are on a time scale that's less than 16 hours, right? Because that doesn't even capture the digestive cycle or the circadian cycle. But we are limited in time resolution due to the experimental limitations. And that's something that we'd be very eager to find out if there are advances in experimental protocols and techniques that would enable us to overcome that hurdle and go to denser time sampling, so we can get a much clearer picture of the dynamics. We don't know about extrapolation. We have not tried to extrapolate. We've been able to interpolate. We have good, positive evidence that that's effective, but extrapolation we have not tried.
Understood. I'll not take much of your time, but yeah.
Sure. Well, thank you, everybody, for your questions. They're very thought-provoking, and it is a pleasure to be here to explain a little bit about our research.
Well, thank you for presenting.
And for answering all of our questions. And, this is wonderful research. We are looking to learn
Thank you very
about
much. Great.
I wanted to ask about the plant.
Yes.
Yeah, but we didn't get to it. Maybe for sure we'll invite Ophelia here, and maybe one day we'll invite Mike.
Yeah.
We're
really
losing you.
With pleasure.
Okay.
It was really grand. Thank you.
Thank you, everybody, and have a great week. And hope everybody sees some exciting scientific presentations over the next couple of weeks. I'm very impressed by what Clubhouse has been able to put together. This is a really nice medium.
Thank you.
Yeah, you should come back.
Yeah, that's a good connect to what comes next. So yeah, follow the club. We'll have more rooms like this. We have tonight Dr. Cotwell talking about high precision measurements of the W boson mass. We'll have on Wednesday Dr. Spurkis talk about rare deep sea brine pools and origins of life research.
Wow.
And
yeah.
Thursday, Dr. Zink about IVF sequencing and how it can predict better the risk of miscarriage. And then we'll have an engineer coming, how he and his team engineered mattresses that kind of regulate the temperature during falling asleep and during the night, which improves the quality of sleep. And quality of sleep is really important, as we learned here in different rooms about dementia and health rooms. We had different sleep rooms. And on Friday, we'll have a team coming here, Dr. Ren and Dr. Cheng, talking about new semiconductors that are better than silicon. Yeah, that's our week. And then next week, we'll have more. So feel free to come back. And thanks. And thanks, everyone, for participating. We really appreciate it.
Great.
Thank you.
Thank you. We'll close the room in 3, 2, 1. Bye, everyone, and thank you.